Today we are going to talk about hippocampus. I want to give a special emphasis on hippocampus in this class, and you will learn why. So our title is What is Hippocampus and Why is it Important? It's famous. It's like a very popular brain structure, and we need to know that. So we are going to talk about the anatomical location of hippocampus. We are going to talk about the basic neuroanatomy of hippocampus. Then we will talk about psychological and physiological conditions relevant to hippocampus. They are not actually separated from each other, you know, physiology and psychology. But to study one phenomenon, you need to observe in different complexity levels. So a psychological phenomenon could be to memorize something, to store something in your long-term memory. But what would be, how it could be actually observed in the physiological level? That's what I mean. And then, of course, as it is already clear, a special emphasis on learning and memory. But of course, we will talk about other psychological conditions related to hippocampus, especially for disease states, disease conditions. Because in the upcoming classes, we will also talk about certain diseases, you know. So here in this figure, you can actually see that this is the 3D view, that where you see actually this purple structure is the hippocampus, all right? So, and it is actually the part of the limbic system. So one important feature of the hippocampus is that it's a part of the limbic system, and limbic system structures which are shown in this figure are altogether involved in certain functions such as motivation, learning, and emotion. So one aspect of hippocampus is this. And structurally, as you have already in the previous class, this is the sagittal section, but if you have the brainstem structures here, they somehow conceal the visualization of the hippocampus. So if we remove the brainstem structures in the second slide, picture or figure, you're going to see actually that is the shape of the hippocampus with the amygdala in the anterior part of it. And of course, we would like to know how hippocampus is actually looking from different angles. And this is this beautiful coronal section, okay? And then where you see here, you see the lateral ventricles. This is actually the anterior horn of the lateral ventricle. Then, you know, it's like a C-shaped, and this is the inferior horn, which is actually lies in the very close proximity with the hippocampus. So hippocampus in the cross-section looks like this, which is actually in the 3D like a C-shape, but if you have a cross-section, it looks like this. So it's the fold of different tissues all together, and it is found in both of the hemispheres, like this, all right? But of course, we would like to know here the structure which is surrounding the hippocampus. For example, here in the, you know, a bit anterior to the hippocampus, you have pararhinal cortex, and then you have entorhinal cortex, which is in the a bit down part, and then it is surrounded to the posterior part by parahippocampal cortex. These structures of the cortical areas are involved in the input-output relationship of hippocampus with other brain regions. And when we would like to have a section, you see here actually the major regions of hippocampus. So it may sound a little too much detail, but it is not, because a lot of very, very important experiments have been conducted in different regions of hippocampus. And those famous experiments, when I need to mention them, sometimes I need to use these terms. That's why you need to be a little familiar. Here you see that this different CA3 region of the hippocampus, CA1 region of the hippocampus, and the dentate gyrus, and then other surrounding entorhinal cortex, parahippocampal cortex. We will see these CA3 dentate gyrus structures in more detail. And of course, I would like to show here in the rat hippocampus. So humans have hippocampus, all the vertebrates have this hippocampal formation. And of course, a lot of important experiments were done, the slices of the rat brain obtained from, which actually have an isolation of the hippocampal slice. And that slice actually here, if you can see, this area is the dendrite gyrus, and here is the CA3, CA2, and CA1 region. And here one prominent cell is granule cells, for example. In the CA area, there are other kind of cells, and one of the most prominent cell type is the principal cells called pyramidal cells. Both granule cells and pyramidal cells are excitatory cells, and actually the way how they are connected with each other have a specific name. For example, when the CA1 pyramidal neuron is projected to the CA3 pyramidal cell, that connection is called Schaffer collateral. And for example, tracts from the entorhinal cortex, so from the other cortical areas, when they actually synapse on the granule cells in the dendrite gyrus, this path is called perforant path. So these are just basic terms regarding the hippocampal connections, and today I will describe one important experiment which actually took place in this connection between these two neurons, which led to the discovery of a very important physiological mechanism called LTP, or long-term potentiation, which is today considered to be the physiological basis of learning and memory. So this is an Egyptian god, and actually if you can see here with these horns, that is actually where the CA, where is that? The corny ammonis is coming from. So the CA regions that we describe here, actually CA1, CA2, CA3, is just because the hippocampus itself is, resembling these horns that somehow made people to give this name because of this old Egyptian god. Okay, so that is the relevance of the CA vocabulary. Okay, actually, you are not responsible for this slide, but I just would like you to show an example of different cells and connectivities and their interactions. Here, for example, in the slide, you see here, let me look at from a little distance where I can see in a better way the different cell types. For example, this one is a granule cell. So when I say granule cell, it's a subtype of a neuron. So it's an excitatory cell. And then you see there is an interneuron. These are usually inhibitory cells. And then here you can see other different kind of cells, but look at the pyramidal cells in the CA3 regions and how the interneurons actually are interconnected with all these different cells. Okay, so there are a lot of different kind of cells. This is just a little example to see that how many different neurons with specific molecular and physiological characteristics are actually found. Even one bit of hippocampus in the dendrite gyrus and CA3 areas. Okay? So, right, so I think now you have some little understanding about what is hippocampus and this, circuitry that involves the CA regions and dendrite gyrus. And now I would like to talk about some very basic features related to hippocampus, especially because it is one of the most studied brain structures in different levels of complexity. So that's my famous term that I used a lot in this class, different levels of complexity. So I want you actually to have an understanding that what the psychological phenomenon is just what maybe we feel, we observe, but if you want to study that in different levels of complexity, there you have to find molecules, there you have to find cells, there you have to find neural circuitry, there you have to find brain imaging scans, all these things, and you have to somehow really have a scientific understanding of this phenomenon, then you have to consider all in an integrated way. That's why sometimes I talk about psychological standpoint, sometimes I go down and talk about molecular and cellular standpoint, because basically it's the same different levels of complexity, and we cannot separate it from each other. And in this course, I want you to understand that these topics related to psychology is actually topics of biology and neurobiology, and we cannot just understand it scientifically if we don't know brain. Okay, so now let's come back again. Why the hippocampus is famous? Because it has been studied in different levels of complexity, meaning that one level of complexity that you study the hippocampal function by means of neuropsychological studies. What are the neuropsychological studies? For example, when a patient has certain brain regions removed, and then as a result of a surgery, for example, and then some behavioral assessments on that case can be conducted. And as you remember with the case of the HM, one of the most famous case studies in the history of neuroscience. And also in this famous study, of course, the medial temporal lobes were removed, in which the hippocampus is also found. And with this famous study, we have actually came to the conclusion that there are multiple memory systems. So the topic of memory, there are multiple memory systems and probably mediated by different brain regions, have been understood first with the studies done by actually Brenda Milner and HM case study. And there the main topic was again hippocampus. So that is one reason why hippocampus is famous. But there are also many other famous studies that were conducted on hippocampus. As I said, LTP, which we will talk today, the long-term potentiation, which is one form of synaptic plasticity, which we are going to talk today, are actually first discovered in hippocampus in animals. So that's why hippocampus is famous. Yes, I talk about that. And of course, hippocampus is associated with many diseases. So any kind of prevalent psychiatric disease, such as schizophrenia, such as depression, you find that actually there is a trouble related to hippocampus. Basically, hippocampal atrophy means that you have the degeneration of the relevant tissue. Okay, you have the loss of the tissue volume for that. And that is hippocampal atrophy has been observed in different diseases, including epilepsy, Alzheimer's disease, depression, and schizophrenia. Here you can see, for example, the comparison of the healthy brain and the brain of an Alzheimer's case, in which actually it's a very severe atrophy, very severe degeneration of the actually not only the hippocampal part, but also the other parts. So, but because basically the Alzheimer's disease is all about just, you know, there is this degeneration of the brain altogether, which causes them certain neurological dysfunctions. But for depression, so in the Alzheimer, the loss of brain volume and the degeneration of tissues is a general globally observed in the brain. But for depression, there was this very specific observation that almost, nearly 20% volume loss has been observed in patients diagnosed with depression. So meaning that somebody who has a official diagnosis of depression, so in the neurological level, their hippocampus may be like having a loss of 20%, almost 20% loss in the volume. So that is here, as you can see in this example, this is the hippocampus of the healthy person, and this is the person which actually is depressed. But of course, this is like a very prominent case. Yes? Yes. What about inorganic depression? Do we have inorganic depression? Depression is organic.
No, I mean, in Russian, we have organic depression. I don't know how to say in English this word.
So,
that causes damage of your
mhm.
brain. But depression and your neurons like
Yes,
stop
working properly.
right.
Yes,
Bipolar people.
yes. Well, I don't know the classification of depression in Russia, but just to give brief information, so depression is today, according to modern medicine, is diagnosed based on the certain guidelines. These guidelines is, for example, Diagnostics and Statistical Manual for the United States and International Classification of Diseases, ICD, for the majority of the countries. So they are also pretty similar with each other. And there, actually, the diagnosis of depression is not really based on this kind of measurements. So nobody is going to scan your brain and look, you have a decrease of your hippocampus, so you might be depressed. No, usually what doctors do, you go to a doctor and then you tell your symptoms. And based on those criteria that I mentioned in those guidelines, like loss of appetite, low mood, inability to sleep or oversleeping, inability to eat or overeating, sorrow, so like a lot, not just because you feel down, but like at least a certain long period, they may diagnose you as depression. But that is actually a topic that we will also talk in the upcoming classes, because that is one problem that actually psychiatry does not have biological markers to diagnose its diseases. Psychiatric classification is based on symptoms. And that really works well in the level of clinics because it's reliable. So what does it mean it's reliable? So if I'm a medical doctor, if I follow that criterion for one patient, another doctor using the same criterion is going to lead or is going to conclude the same diagnosis for the same person. That means the reliability, and that is very useful for clinics. However, it doesn't have validity. That means it is not based on the biological criteria, and that is one of the current problems in psychiatric disorders. And that causes also a lot of issues regarding the treatment, which we will talk in the upcoming classes. But in this case, what I'm trying to explain that there is such a difference. Hippocampal atrophy was observed in the majority of the depressed patients. But when I say depressed, I really do not discriminate any kind of organic or whatever depression is, because if your behavior is somehow distorted in a certain way, that means there is something going wrong in your brain. So we already, that's why the course is based on that idea. Otherwise, we wouldn't study brain in order to understand behavior. And there are a lot of actually scientific evidences showing that this assumption is correct. That for that, you need to had to come to my first class, which you missed.
So you mean like biology is the main thing in psychiatry?
Psychiatry is a division of medicine, and the main medicine is an applied field of biology.
I hate psychoanalysis, but by all my heart.
No, I like what I like is I think psychoanalysis is interesting too, but that shouldn't protect us to study physiological aspects. And there are a lot of there are some scientific data supporting psychoanalysis also. Certain features of the psychoanalytic theory can be actually shown by some modern neuroscience experiments. I also talk about psychoanalysis, but it was in the first class, so it will be open, you can watch. Okay. So basically here we mention, I think that hippocampus atrophy is actually correlated with diseases, including depression, okay? So that is, and then the question is, okay, there is a correlation with hippocampal atrophy, but is it is it a cause of the disease? That means people are depressed because their hippocampal volume is decreased? Or is it actually the result of the disease? Like people have some problems in the brain, and that one of the so those problems will also end up actually decrease in the hippocampus. So is it the result or is it the cause? We don't know very well. But what we know, for example, if there is a loss of tissue here, for whatever the reason, so any effect that reverses this situation would be good to increase the tissue. And actually, the born or formation of new neurons is a process called neurogenesis. And people found that, for example, certain antidepressants increase the formation of new neurons in the hippocampus. Selective serotonin reuptake inhibitors. I don't want to give the commercial name, but I can tell you after the class, okay? And also, but interestingly, exercise. So they did this kind of studies in the mice, and chronic regular exercise showed the similar effects like antidepressant effect, causing the increase in the formation of new neurons in the hippocampus on animals. So like the exercise seems like very good, not only for general health, but not only for brain health, but specifically for the depressed people who have actually very decreased hippocampus volume that can be maybe facilitated by exercise, because exercise, at least in animals, have been shown to increase the neurogenesis, formation of new neurons in hippocampus. So that's why we start to think that, okay, hippocampus may be related to some certain forms of depression. But of course, there are a lot of questions in that aspect, and that is not our topic today. That's why I want you now to focus on. So this slide was just shown to you that hippocampus is related to many diseases, including depression. That's it. We will talk about depression later. Another important aspect why I wanted to talk about hippocampus in this course, because LTP was discovered in hippocampus, and I don't want anyone in this class to graduate from this course without knowing LTP, okay? I sometimes meet psychology students and I ask them, what is LTP? And they never heard, and I'm so upset. That means they are actually missing a century. So this is I'm serious about this, and I don't want you to be in that position, although even you are not psychology students. So, and LTP was actually discovered already in 1970s, ladies and gentlemen, so it's not even in the 21st century, okay? So, but before that, we may need to know we just need to remember this slide in which we have the, from the perforant path, you have this to the granule cells, you have the perforant path from the entorhinal cortex, and then you have the CA3, CA1, which is the Schaffer collateral, in which the LTP was discovered. So in the Schaffer collateral, all you have to think of the two neurons that are connected with each other. And then you have the slice, and you want to study the behavior, physiological behavior of these neurons in the physiological conditions under the microscope. And that was actually done by two scientists, Willis and Lomo, that I will talk in more detail. For the moment, all you have to remember that this famous long-term potentiation, which we will talk in a few minutes, is discovered in hippocampus. Why LTP or long-term potentiation is important? Because today, majority of the scientific circles think LTP is a mechanism of neuroplasticity which accounts for some forms of learning memory, okay? And that is what we have to know. This is just an example of different cells, but I don't want to go there. But now, as you already understand the importance of hippocampus for, diseases and whatever, but actually hippocampus is especially famous for being important for learning, memory, and spatial navigation. But when we say memory, memory is actually not just a unitary process. Memory is not just one thing. Let's define learning is a process that will modify a subsequent behavior as a result of experience. And memory is the ability to remember past experiences. So basically, memory is actually the trace what you have learned, trace of things that you have learned. And they are actually pretty related to each other. So memory, of course, is essential for all learning. What if the point, if just you forget everything what you learn, there is no sense. But so memory depends on learning also. I mean, if you don't learn anything, you cannot memorize. Learning also depends on memory. How do you think learning can be dependent on memory? Yeah, certain things require association of previously learned things. You know, actually, majority of the learning that you do in the university is required previously learned things and their association with the newly formed learned memorizations of the relationships. That's why both of them are pretty related to each other, but they are different. So actually, if you would like to talk about the memory, the first thing you need to know, as I already said, it's not a unitary process. That means that there are different types of memory, and I'm not just saying because I assume. Whatever I say in this class are told because there are experimental evidence. Okay, that's very, very important. To make theories is difficult, but not as difficult as to prove them with experiments. So if I say there are different types of memory because there are experimental results showing us, and probably they are mediated by different systems, and one actually indication of that was the study done on HM, the case study in which the medial temporal lobe was removed. If you actually are curious about this topic, I actually would promote you to read that paper related to the studies of HM, or something related to a text explaining what has been done. You will see that actually the HM study allowed us to understand there are different memory systems. So this is a slide that I just took from Nature Reviews of Neuroscience. So Nature Reviews of Neuroscience is one of the important journals that you may like to follow if you are interested in brain aspects of psychology. Here you see actually there are different type of memory, long-term and short-term. Now let's focus on the long-term memory. Then you are going to see that there is declarative memory and there is non-declarative memory. Sometimes it's called explicit memory and implicit memory. And actually, this form of long-term memory can be perceived as conscious memory and unconscious memorization. So certain things can be learned consciously, but their memorization is not necessarily supposed to happen consciously. For example, skills. So you can learn how to drive a car, but when you drive the car, that procedure or that skill of driving a car does not require you to think intentionally and consciously. When you really learn it well, you are just going to be in the automatical mode. You will just drive the car, you will touch the gear shift and everything automatically, or biking or swimming. So these are the skills that may require conscious learning, but their memorization, memory retrieval is automatic. It's not really consciously happening. On the other hand, there is, in the declarative memory, most of the time, for example, there is this episodic memory and semantic memory. I wrote all the definitions here for you underneath to check at home. But basically, when you have the episodic memory, you just remember things in association in a spatio-temporal context. For example, what I was doing last year these times. So you see, I have to think about something long-term. I have to think about the time. I have to think about the context, what I was doing, where I was, in which geographic location. Probably I was not in Russia at that time. What I was doing exactly, all these things. On the other hand, in the semantic memory, I don't know, the general knowledge, the general facts that are not really associated with a specific context are taken as semantic memory. Just give an example. Think of a cat and a mouse. So what would be the reaction of cat to the mouse? To attack, right? Tom and Jerry. So normally everybody would know, at least if the cat is not trained, typically a cat will attack the mouse. And the mouse naturally will always escape from the cat, right? But you never remember how do you get this information? What was the context? So this is just general facts that you learn. Okay, so these are these different kind of learnings, and hippocampus associated learning and memory is actually in this camp. Okay, so when I say learning and memory, I'm talking about for hippocampus in this area. And now, if we are talking about the memory and there are different, the fact that there are different memory systems, then we have to think that which kind of these different memories that I have shown in the previous slides are known to be associated by which brain regions. And then we come to the conclusion that, for example, the prefrontal cortex is more or less related to actually the working memory. Working memory is a special form of short-term memory. And procedural memory, which is related to unconscious learning and memory, is actually mostly mediated by cerebellum and striatum, not by hippocampus. And amygdala is more expected to be related with emotional memory. And look at medial temporal lobe, together with hippocampus, is involved in the long-term semantic and episodic memory. Okay? Here is an example to show you how different brain regions are involved. In this figure, there is this brain scan of an individual that the person is asked to remember the face of somebody, I don't know, the face of your mom, the face of your friend, the face of your first love or crush, but maybe that will make more different areas to be activated at the same time. So let's don't go there. So when somebody holds the face of somebody on the mind working memory, usually the areas in the prefrontal cortex are active. This is the horizontal scan. Mm-hmm. So obviously there's a degeneration in the associated brain areas.
These sections are not working, right?
I don't know if specifically that sections, probably some other sections too. I don't know. Okay, now actually how I would like to show how hippocampus is connected with different brain areas. Because as you already know, I mean, in the simple slide where you have the sagittal section, as you already know that this is not as simple as this. There are so many different structures. So the hippocampus is here. And actually, these are the major connections that is hippocampus interacting. So these connections have to be perceived in two levels. The cortical connections, the subcortical connections. Here you see on the top, the subcortical connections, thalamus, hypothalamus, basal brain. Especially you see thalamus and hypothalamus are actually interacting with the hippocampus by fornix. And then you see the amygdala, on the other hand, is directly connected to the hippocampus. So there is like direct connection between hippocampus and amygdala. That is very interesting. And that is, as we already know, both of them are found in the limbic system. In the cortical level, hippocampus is interacting via entorhinal cortex with frontal lobe, with temporal lobe, and parietal lobe. Okay, so there is cortical interactions via entorhinal cortex. There is subcortical interactions, especially to the thalamus and, hypothalamus, basal forebrain via the fornix. So cortical, subcortical structures, and hippocampus is just, interacting in both ways. Okay, so now we can talk a little more about LTP, the hippocampus, because it was discovered there. And what is actually the LTP? first. But before LTP, I have to tell a little about the history of this idea. What is idea here? What actually people are trying to do in this experiment? What actually inspired, what kind of things or ideas inspired them to do some weird experiment in a specific you know, if you don't know the background idea, you cannot, it is very important to, you know, see the background idea to understand the basis of the hypothesis. So always the background idea or background evidences actually inspire people to make an hypothesis to test. So almost 100 years ago, as I already mentioned in one of my classes, Ramon Cajal postulated that memory is actually should be stored in the synaptic connections as a strengthening of the synaptic connections. So almost more than 100 years ago, he proposed that idea. And the idea was somewhere, okay? But of course, people are reading, people are investigating, people are working on that. And there were some other postulations also based on the initial proposal of Cajal. So this idea that memory can actually be stored in the increased connectivity of the synapses connections in specific brain regions have become very popular. But, you know, to actually prove it, the point is how to prove it. That's what we need. So without proof, it doesn't work. Without proof, we can be like psychoanalysis. So we need proof. For that reason, no, I like psychoanalysis. So for that reason here, so what they thought, how come actually a synaptic connection can be stronger? What would cause a synaptic connection in the physiological level can be increased, can be stronger? It is very simple. If you make some connection active, let's assume, it is expected to be stronger. Think about your muscles. If you use a muscle all the time, even with lifts, it will become stronger, right? So maybe something like how can actually this connection between synapses can become stronger? Maybe if the connectivity, the signal transmission between those synapses is stimulated more than a threshold, that is, the synapses are overactive, and maybe as a result of that, those synapses are going to get stronger. So that was the initial idea. And for that, they actually, you know, in this experiment, which was done in the 1970s by Bliss and Lomo, they isolated the hippocampal slice, and then they just here in the Schaffer collateral, which you remember from the previous slide, that they actually have two kind of electrodes. One is actually here. Here you see. So this is an input electrode, a microelectrode, that they can stimulate one neuron. So if two neurons are connected, if you stimulate one neuron, you will be able to measure the response of the other neuron. So they, in the Schaffer collateral, introduced one stimulating electrode, and then they introduced in the CA1 of the Schaffer collateral another electrode, which is a recording electrode. So there are two microelectrodes, stimulating electrode and recording electrode. And then they start to stimulate the first neuron with different kind of frequencies. And then they wanted to measure the response of the pyramidal cell in the CA1 region by recording electrode. Okay, and then they try to find out a relationship between them. And what they have found actually that if the neuron is stimulated with the stimulating electrode, okay, there is a response of the, let me show. In a more simple way than this. So let's say you measure, this is the recording electrode, and stimulating electrode and recording electrode. Okay, let's say there is here this amount of stimulus or current injected to the first neuron in the Schaffer collateral, and then in the recording electrode, such a kind of response is recorded. But if the frequency, so then there is another current is injected and the neuron responds in the same way. But if the frequency of the stimulation is not this, but if it is like very, very much frequent, okay, much frequently given, then the response of the neuron as recorded by the recording electron is actually not like this anymore, but it is like this. So the current recorded in the second electrode is increasing as a result of the frequent stimulation, high frequency stimulation. So high frequency stimulation of the first neuron will cause an increase in the response of the second neuron. So that means that if the connectivity between two neurons are very frequent, then the second neuron will eventually increase in terms of its response level. And that can be measured by the electrode recording, okay? And then, so here the idea is that if there is a stimulation coming to the specific neural circuitry at above a certain threshold, which is high frequency stimulation, that will cause the responding neuron to give higher response, as it is shown here with my explanation. So actually, that is actually how they have actually found this phenomenon. And later, the later studies have led to the identification that such a event, that the potentiation of the response of the target neuron or second neuron is actually happening because the synapses becoming stronger as a result of insertion of new proteins into the postsynaptic cell. So there is, as you remember, there was a presynaptic site and postsynaptic site in a cell. And there are certain receptors that are found in the postsynaptic cell. And how does it happen that suddenly there is a certain profile of the postsynaptic response? How come it is more? So they found out that because more postsynaptic receptors are actually placed as a result of this extreme activity of that synapse. And this kind of phenomenon is called long-term potentiation because synapses are potentiated. And that happens because of activity, and that's why it is called a form of activity-dependent synaptic plasticity. Here, in this figure, you can actually understand that we are in the level of decode the memory process in terms of molecular cascades. So that phenomenon in the physiological level can be actually understood in the molecular level. What happens in this, it is explained in this slide. And that is basically what we have in the slide is the following. This is the first neuron in the Schaffer collateral, okay? In which the stimulating electrode is inserted. This is actually the second neuron in the Schaffer collateral CA1 region, in which the recording electrode was inserted in the experimental level, in the experiment. And then, as a result of the high-frequency stimulus, what happens? You know what happens? I will explain. So this is, let's say, this Schaffer collateral neural connections are glutamatergic pyramidal neurons. So that means that this presynaptic neuron is a glutamatergic neuron, precisely matching with the glutamate receptors that are found in the postsynaptic cell. Let me get my stick. I forget. So in the postsynaptic cell, there are different receptors, but these all are called glutamate receptors, but there are different subtypes of glutamate receptors, and that is the point here. The point that why high-frequency stimulation is required in order to actually change the synaptic strength. So This is NMDA receptors and AMPA receptors. These are two types of glutamate receptors. Both of receptors are permeable to positively charged ions if glutamate is bound to them. However, there is a difference between NMDA and AMPA type of glutamate receptors. In the AMPA receptors, this is the glutamate. Once the glutamate is bound, okay, the sodium ions will have an influx to the cell. Okay, so for them to get active and to allow the influx of sodium ions into the cell, they need glutamate bound to the AMPA receptors. However, glutamate receptors are not so easy. For glutamate receptors to get activated, they do not only need actually glutamate, but also they need a kind of you know, there is this magnesium here. I just don't want to go there too much, but there is this magnesium block here that somehow it blocks the channel. And even there is a glutamate binding because of the magnesium block that is found in the gate of the receptor, it will not get activated. The only way to make this receptor active requires both removal of the magnesium and bound of glutamate at the same time. And that can only be achieved if this neuron is, as a result of the stimulation of these receptors, is depolarized already, because sodium, if sodium goes inside the cell, it will cause actually, you know, the postsynaptic neuron to get excited at a certain stage. And that is a slight depolarization. And that depolarization will help the removal of magnesium block. If time there is glutamate bound to the postsynaptic receptor, that will cause actually positively charged ions to get into the cell. But in this case, there is a difference in terms of the ions to which are permeable to glutamate receptors. And that is, for AMPA receptors, it is sodium. For NMDA receptors, it is sodium and calcium. So the calcium entry here is the key. So calcium maybe, you know, for calcium for the body appears to be important for the bones, but actually for the cells, for the brain, for memory, calcium is a key secondary messenger. We call them as secondary messenger because calcium has more to do than just changing the membrane potential. Calcium has ability to trigger certain molecular cascades. If I'm going from here to this direction, what do you understand? Where does it go? So if this is a synapse, but this is the postsynapse, and where does it go like this? Where does it, where? Where does it go? Exactly, it goes to soma, and what do we have in soma? Nucleus and genetic material. So calcium actually triggers all these events downstream to the nucleus of the cell in which there are genes, and calcium for that reason has the capability to turn on and off genetic expression. That's why calcium entry to the cell is tightly regulated. Calcium is just, you know, important dude here. He or she will not enter to the neuron easily. She will not, or he will not bother himself easily. That requires already enough depolarization plus glutamate at the same time, because calcium then is actually going to affect on the genetic level. The calcium will going to turn on genes, turn off genes, and that will actually cause the insertion of new receptors eventually. There are some other molecular cascades here, but our course topic is not molecular neurobiology. These are the topics of molecular neurobiology. That's why I don't want to go there in detail. But if you guys are interested in molecular biology, just tell me, then I can actually make another elective for that. But basically, what I'm trying to say here, that this LTP happens because this connectivity between these two neurons in the synapse is changing. And how is it changing? It can change by two ways. It is only changed if the molecular composition of these two sites are changing. And that can happen by two ways. Either the existing ones, for example, these are the existing receptors, but these existing receptors can also be modified. Their structures can be modified by, for example, binding of some molecules such as phosphates. Okay, phosphates can change the channel kinetics, for example. How long this channel is going to stay open? How long it will allow, until when it will allow the influx of ions, for example. All these factors can be modulated. So one way of changing the synaptic strength is actually to modify the existing receptors. And that is actually, for example, to potentiate the kinetics of this one. That will cause a stronger interaction between them. Another way can happen by genetic factors. If calcium will lead to the activation of certain genes, which will actually allow the insertion of new receptors. So the existing one can be potentiated, new receptors or proteins can be inserted, and by this way, a synapse can become stronger. Okay, so actually this idea was already a very elegant experiment, was actually already showing this phenomenon in a rather simple organism. That organism did not have a hippocampus, but you know, in science, we usually get inspiration from simpler organisms because we are reductionists. We want to reduce the complex system into rather simple systems in order to understand the very fundamental principles. And one elegant way to do that is look at this organism, the sea slug. So these sea slugs were chosen to study one of the most interesting studies in the field of neuroscience because they have a simple nervous system, but there are many other organisms with simple nervous system. Plus, they have actually, their neurons are very big, the ones that we have in the vertebrates, and that will allow a very practical way of studying them. And they can even model certain type of, you know, simple learning. Think of Pavlovian conditioning learnings. What about all the animals have these kind of simple learnings that can be modeled? And then you can actually isolate the nervous system, which is actually modified as a result of conditioning, and study all the molecular and physiological changes compared to the control organisms, and you just find out what is happening there, and you can understand even the simple learning is actually happening as a result of the change in the synaptic connections. And that was done already in 2000 with this elegant experiment. Here in this experimental paradigm, I would like to show you that this is actually, this is the head of the sea slug, and this is the tail, okay? And it has this area called siphon and the gill, all right? And this animal has a specific reflex. For example, if you stimulate the siphon with physical stimulation, tactile stimulus, or you can actually just, make a water pressure. And of course, that is sensed as a threat by animal, and the animal is going to, withdraw. Look at, this is the first stage, and this is the second stage after the tactile stimulus. The animal is going to withdraw its gill and close itself. But if this stimulation is by a few times, after a while, animal gets habituated. There will be the case of habituation, okay? Habituation. So the animal actually, will actually animal do not respond to that as much as before. That's why it is actually, this is the first stage and this is like second. But then we can sensitize the response of the animal if we pair the siphon stimulation with an electrical shock into the tail. So when the siphon stimulation is paired with the tail shock, look at the animal, how the actually gill withdrawal is so prominent compared to the first stage or even the habituated state. Okay, so they then isolated, so then the animal actually associated the tail stimulation with the gill withdrawal reflex. Okay, and then once those neural connections associated with this behavior, the gill withdrawal reflex, is isolated and studied, what they found is the following. Here in this slide, you see the neurons that are found in the gill withdrawal reflex, and you have two cases, the control animal and the animal in which there is a sensitization that the animal is associated, the tail electrical response with the gill withdrawal reflex. And here you see the siphon is here, tail is here, and the gill muscle is here. The siphon has actually is the one in which the sensory neuron is emerging, right? The sensory neurons receive information from the external stimuli and then transmit that information to the other parts of the nervous system in each kind of organisms, including this organism. So when you have the sensory neuron is connected with the motor neuron. So the stimulation of the siphon in the sensory neuron will cause the gill muscle to be withdrawn, because this neuron is going to activate the motor neuron, which will actually activate the muscle cell. Okay? But if you pair this gill withdrawal reflex with a tail, so you give an electric shock to the tail of the animal, tail of the organism, and then what are you going to see? At least what they saw when they isolated the system. And then there is this neuron, which is actually a neuron that is a serotonergic neuron by nature, and you already know what is serotonergic neurons, as we already have in the human and vertebrate brain. These organisms also have those modulatory neurons that have the serotonin as a neurotransmitter. So that is their connectivity point here. And if we make it a big zoom, what do we have here? Okay, so we have two cases. In the first case, there is a little brief electrical shock. In the second case, there is a strong electrical shock, which is like which actually caused the animal to get sensitization. So what happens here in the first case? So when the tail shock, so the neural circuitry associated with the brief tail electrical shock has shown that this modulatory neuron is going to cause an activation of a specific protein called PKA, and that protein is actually facilitate the release of this neurotransmitter from this neuron to this neuron. So there will be some facilitation of the interaction of these two. However, if the electrical tail shock is really strong, then there are more prominent effects observed in the cellular connectivity. And that connectivity is actually, to say the last thing first, because of the genetic activation that caused the formation of new proteins inserted into the cells, or sometimes, as you see in this case, the formation of new connection between two neurons. So what happens in this case that this PKA is also going to actually, it is more potent in this case, and it will actually activate some downstream factors that are actually found in the nucleus, and the genes are going to get activated, which will cause the formation of even new synapses, not the potentiation of an existing synapse, but formation of new connections between the sensory neuron and motor neuron of the C-slux neural circuitry associated with this gill withdrawal reflex. So by basic isolation of the nervous system, it is actually showing the gill withdrawal reflex sensitization. They have found these factors that are experimentally shown and proven. So as a result of that, today we can say that the basic learning, at least in this basic system, is already mediated by this kind of formation of new synapses that are triggered by the modification of the gene expression. And that requires already, more activity, so that is a form of activity-dependent synaptic plasticity. In this case, there are new synapses formed. That's why I would say neuroplasticity. Neuroplasticity is a general term. Synaptic plasticity is modification of synapses which are already existing. But in this case, the new synapses form. That's why I don't want to call synaptic plasticity, but neuroplasticity. So here I especially would like to give a definition of certain terms. Where is my... No, it's not here. But to summarize, long-term potentiation is a process that has been already discovered in the 1970s in the hippocampus. But then I actually mentioned some other studies that was done in the sea slug. So we have like a converging results regarding the molecular, physiological, and behavioral aspects of memory. Okay? And all these results converge on the idea that there is this mechanism of neuroplasticity. Neuroplasticity can be defined as the ability of the change of the nervous system as a result of activity. And that happens during health, that happens during disease. So neuroplasticity is a natural phenomenon, but it is also in the pathological states, it can be also observed. But for learning and memory, at least for the declarative memory, for hippocampus, today we think that it happens as a result of synaptic plasticity, which is one form of neuroplasticity, in which the synapses get stronger. Is there also opposite form of synaptic plasticity for the case of long-term potentiation? Yes, there is also long-term depression. Some synapses can become less active, which is the opposite of long-term potentiation, which is called LTD, long-term depression. So we actually observe in different hippocampal cells and also in different cortical areas, different form of neuroplasticity for certain forms of learning and memory. But basically, neuroplasticity is thought to be the mechanism for learning and memory, at least some forms of it. But in general, neuroplasticity can be also observed in different disease states, in different developmental stages, in different contexts. But for the case of hippocampus and learning and memory, so one of the important things that we know is with the support from the experimental evidences is that, yeah, the learning and memory occurs as a result of the activity-dependent synaptic plasticity, which cause actually the modification of genetic expression, and that will actually impact on the synaptic strength by the insertion of new proteins to the synapses. And that has been observed in the vertebrates, that has been observed in the other organisms as we already described. So we have like a pretty converging results on understanding the mechanism of learning and memory in different systems, including hippocampus.